Alright guys, so today's video is going to be about a nerve itself and how it works. So how it's able to actually transmit a message down the length of the nerve to the either muscle or gland that it's headed to. So we're going to start with this diagram here, and we're going to kind of label some parts and talk about what they do. So the first part is going to be these little tree branch looking ones up at the top near the cell body. Those are what are called our dendrites. Dendrites are going to receive information. So dendrites will receive the stimulus. So they'll receive the message that's coming into that nerve or the neuron. This middle part right here, this is going to be the cell body of the neuron. So this whole big thing is one neuron. So it's one big giant cell. So that cell body, you'll see there, contains lots of organelles, just like a typical everyday cell has. It has a nucleus in it. It'll have some mitochondria in it, endoplasmic reticulum, all those kind of typical organs. In addition, it will have something unique called a nasal body. A nasal body is going to contain lots of RNA that is going to be concentrated here in the cell body. You can see all these little blue packets here. These are all nasal bodies inside of your cell body there. There's lots of mitochondria inside that cell body. If you remember from freshman biology, mitochondria make ATP, okay? And the neuron needs to make lots of ATP. So there are lots of mitochondria there in that cell body. And these neuron cell bodies, they do not divide. So unlike skin cells and bone cells that are constantly changing and dividing and repairing and replacing, neuron cells do not do that. So they do not divide. This last part here of our neuron here, that's our axon. And if you notice the arrows on the axon here, so this is going to take the message away from the cell body. So the dendrites are receiving the stimulus, taking it toward the cell body. The axon is going to be taking it away from the cell body. Where that axon forms, right here, where the neuron cell body kind of starts to narrow down to the axon here, that is what's called the Axon Hillock. And that's where the electrical impulse that's going to travel down the axon, that's where it's really going to start to form. Down here at the end, these are what form our synapse down here. Those are the axon terminals. And we'll look at a blown up picture of a synapse in one of those in just a minute. So let's look a little closer at this axon structure while we're on this larger diagram here. So in the axon here, that's our myelin. If you remember from the previous video, that's part of the myelin sheath. Remember, it's formed by those Schwann cells. And that myelin is made out of a lipid, which is a fat, which acts as an insulator. So if you think to your physics classes or IPC or middle school, right, an insulator means that no electricity is going to get through that area. So actually, that's going to make the message, the electrical impulse, be conducted even faster than if it's unmyelinated. We'll talk about why that's so. Unmyelins are called the node. They're named after a guy who was French, so it's pronounced Ranvier. So the node of Ranvier, those are unmyelinated areas, so they are not insulated, meaning they're actually going to be able to conduct those electrical impulses. So again, remember, these are your notes. Write down what you need. So if you need to write all of that out, right, unmyelinated, therefore there's no lipid, no insulator, messages can be conducted. Write all of that down so that you'll remember it when you go to study. So axons that are myelinated are what are called our white matter. So these are myelinated axons. Axons that are unmyelinated would be part of the gray matter. So this is unmyelinated. White matter actually has messages that travel faster than gray matter because of the myelin, because of the insulator. So this is a faster type of conduction. And that is going to be faster because the message can actually jump. Okay? It'll jump from node to node like this, instead of having to travel through every single bit like it would on a gray matter or an unmyelinated neuron. And so since it can jump or leap from node to node, the message will actually travel faster, right? Think about if you were running a race and you were allowed to jump or leap versus somebody else whose feet had to touch every inch of the ground from the start to the finish. So the person that was allowed to jump or leap, they're going to be able to go faster. So the white matter with those myelinated neurons, their messages can travel even faster because of that leaping. That leaping or jumping is called saltatory conduction. So that saltatory conduction is going to occur on that white matter. And it's that leaping or jumping of that electrical impulse over those insulated areas. The unmyelinated neurons do what's called continuous propagation. So that's happening on that gray matter. And it is basically every inch of the membrane is going to have to conduct the message, which is going to slow it down and make it take a little bit longer. In addition to whether myelin is present or not, the diameter of the axon can also determine speed of conduction. The greater the diameter, the faster the speed of conduction. So your two big things are going to be whether it's myelinated or not. Myelin will conduct faster through that saltatory leaping conduction, and then diameter. So how big around that the axon is. The bigger around it is, the faster the speed of conduction it will have. All right, so let's take a look at a zoomed in view of a synapse. So these are the axon terminals that we pointed out on our, down there. These guys right here that we pointed out on the other diagram that was starting to look a little bit messy. So these are the axon terminals. They're going to branch out and they're going to go to the various cells that the nerve is going to innervate or control. So these axon terminals are going to be full of neurotransmitters, full of vesicles that are contained neurotransmitters. So if you're looking at this diagram here, the pink part is the actual axon terminal. We've zoomed in. You can see where you've zoomed in on this part here. So these synaptic vesicles that are here at the end of the axon terminal, those are going to contain neurotransmitters. If you think back to muscles, we talked about how at the neuromuscular junction, the neurons release acetylcholine. So that's where the acetylcholine would be stored in these synaptic vesicles. And when the signal comes down the axon terminal, that'll trigger the release of that acetylcholine from the synaptic vesicle there. The rest of the synapse is going to be made of the presynaptic membrane. Presynaptic, so before the synapse. So that presynaptic membrane is going to be part of the actual axon. It's going to be on the axon terminal. So that's going to be this part of the membrane here. And then our postsynaptic membrane, that postsynaptic membrane will be after the synapse. So that postsynaptic membrane will be on the gland, on the muscle, on whatever effector organ, on another nerve, on whatever that nerve is trying to stimulate. Those are your big ones, gland, muscle, or another nerve. The synaptic cleft is the little space between the axon terminal and the muscle, gland, or nerve cell on the other side. It is going to be full of fluid, not water, but it's going to be full of interstitial fluid. Those neurotransmitters, they need to be able to float across the interstitial fluid there. And remember, all of our cells need to be bathed in a fluid. So that synaptic cleft there is full of interstitial fluid. And that's going to be kind of the basis of our synapse. So the message, the stimulus is going to come down that axon, release the neurotransmitters. neurotransmitters will come across to the postsynaptic membrane, and then they'll hopefully trigger an effect in the effector organ. Okay, so we're going to use this graph for a lot, so you may not want to write too big each time we talk about something here. So we're going to start here with what's called a resting potential. You'll see on this graph here, I've got membrane potential measured in millivolts. So basically this graph is representing electric activity. So we'll see the resting potential sits here at about -70. So what that means is there's a difference between the inside and the outside of the cell. So we're going to call this the outside. We're going to call this the inside. And at rest, the cell has whole bunches of sodium do a combo of like pictures and we're understanding what's happening. So sodium and potassium ions are both positively charged. Okay, so that seems kind of weird that we'd have a difference between positive and negative charge here. If I'm talking about the concentration of positive charge on the outside and a positive charge on the inside. However, on the inside of the cell, you also have these big proteins. And those proteins, they're going to have a negative charge. And that's going to offset that positive charge. It's going to offset it and then some. So when we see that -70 resting membrane potential, what that means is that at rest, the inside of the nerve is sitting at about 70 millivolts less than less than neutral, essentially less than them having an equal charge. So the inside is more, essentially what this means is that the inside is going to be more negative than the outside. That's what that -70 millivolts there is going to mean. So at rest, because the inside is more negative than the outside, and there's a difference between the two charges, we would say that the neuron is polarized. It's polar, it has a charge. If you remember talking about water, maybe your freshman year or chemistry or polar molecules, right? That means they have a charge. So the membrane itself has a charge because there's a difference between the outside and the inside, the electrical charges. So when we get to the next step, so this was kind of our resting potential here, we get to our next step, which is going to be depolarization. So if I depolarize something, I'm getting rid of that charge. So what happens on the next step here is these sodium channels are going to open up on the membrane. And so now we've got all these sodium ions that can come inside of the cell. So if I have a bunch of positive ions coming in, there's my big giant negative proteins. If I have a bunch of positive ions coming into the cell, well, that's going to take this -70 and it's going to bring it up, right? It's going to bring it more positive. It's going to bring it closer to 0, which you can see 0 line here. Depolarization actually crosses 0 and makes it become more positive on the inside than on the outside. So when this depolarization happens, if it is strong enough, if enough sodium comes in to cross threshold, you see threshold down here, okay, then an action potential will form. So maybe you only get enough, release enough neurotransmitter to open one sodium channel. Maybe then we don't reach threshold. We don't have enough sodium come in and change the charge enough. But if enough, and so these are opened by, let me go over here, we can see it better. These channels here are opened by the neurotransmitter stimulus. So if we're talking about a neuromuscular junction, right, when that acetylcholine is released and goes across to that postsynaptic membrane, that neurotransmitter causes the sodium channels to open up. So if enough sodium rushes in that the cell crosses threshold, in this case you'll see it's -55, so it's not that much, an action potential will form. That's what that word says. Sorry, it's so messy, guys. So an action potential will form. Action potentials form on what we call an all or none principle. Similar to muscle contractions, right? Where I either got a muscle contraction or I didn't. So I either get an action potential or I do not. It's either enough of a depolarization that threshold is reached, and so I get an action potential, or I didn't cross threshold and I don't get an action potential, all or none. So once that action potential is formed, it will be the same size regardless of how high up the depolarization goes. Again, similar to muscle contractions, once that muscle contraction was triggered, it would be the same strength regardless of how much stimulus is given. So that's where that all part comes from, right? I'm either getting a full-blown action potential or I'm getting nothing at all. And then that will send the action potential traveling down. If I have this big long neuron here, that starts sending the action potential traveling down the neuron. I start the process and it starts moving down the neuron. So you'll notice our next step here is the repolarization, right? If I'm going to repolarize something, I'm going to bring it back to being polarized. And you'll see that my electrical activity here is coming back down past zero, back down to that resting membrane potential, that charged resting membrane potential. So during repolarization, now instead of our sodium channels opening, now we get our potassium channels open. And so the potassium is going to start going out of the cell. While this is a different ion, it still has that positive charge. So you'll notice I didn't talk about chemicals here. I'm only focused on the charges. So I'm going to reset those charges where I'm going to get positives on the outside, more negatives on the inside, more positives on the outside. And so I am repolarizing that cell, bringing back that charge between the two sides of the membrane. During this time, as it's repolarizing, you'll see sometimes it'll go down all the way down to this hyperpolarization phase where it went past, right, our resting membrane potential. So it went more than or past it. It'll go into a refractory period. During a refractory period, it cannot form an action potential. Basically, it cannot receive a stimulus because during this refractory period, the nerve cell needs to be reset so that potassium is on the inside and sodium is on the outside. No, I do not know why sodium just doesn't go back and forth. Okay, so what will happen is this is where our sodium-potassium pump will come into play because I'm going to need to physically use ATP. Remember we said those the cell bodies of the neuron had lots of mitochondria to make lots of ATP because they need to power the sodium-potassium pump. So that is going to reset our nerve completely back to its original state that we had over here with potassium inside and sodium on the outside. I fixed the charge during the repolarization phase by pumping that by sending that potassium out, but now I need to reset the chemicals. And so I pump the sodium out and the potassium back in. We're going to do a manipulative where you're going to actually push sodium out, bring potassium in, bring potassium out, pump them back and forth so that you can kind of really see this happening and use this vocabulary. Okay, so let's look at some different types of nerves and then we'll wrap up. Before we move on to this functional classification of nerves, I do want to take just a second to talk about neurotransmitters. It was just getting a little messy on that last diagram. So our neurotransmitters, they can be excitatory, so they can either cause stimulus, right, cause things to happen, or they can be inhibitory. They can stop stimulus from happening, stop action potentials from occurring or from happening. So neurotransmitters can be excitatory or inhibitory, and after a while, enzymes will break them down. They're not going to be around forever. That's why we don't have, hopefully, ideally, spastic muscle contractions when you tell a muscle to contract, because the enzymes will come in and they'll break that acetylcholine down so it doesn't keep sending the signal. So neurotransmitters, again, both either excitatory or inhibitory, regardless of type, enzymes will come break them down so that stimulus isn't happening permanently. So some functional nerve classifications. So they're functional, what they do. So we have what are called afferent nerves. So afferent nerves are also sometimes called sensory nerves. They're taking information to the central nervous system. So they're receiving information down at the ends, at these dendrites. The message is going to travel down the axon back to the central nervous system. And then your efferent nerves. Those are what we call our motor neurons. They're going to take information away from the CNS. So they're going to take that information out to the effectors, to the muscles and glands, from the central nervous system out to the effectors that will actually do a job. Sometimes you will have nerves located within the central nervous system. They're called association or interneurons. Inter means between. So these association or interneurons are between other neurons. They are located completely within the CNS. So completely within the brain, completely within the spinal cord. So now we're going to look at our structural classification of nerves, so how they're physically built. We're going to look at just these couple of examples and we'll be all set. So our bipolar neuron is going to, bi means two, right? So it's going to have two processes that come off of it. So they're going to have one main dendrite and one axon. So there's our two processes. You can find examples of these in the eyes and in the nose. The second one here is this unipolar. Uni is one. There's the process right there. So that's the one process that's coming off of this neuron. It'll have dendrites located at one end of that one process, and then the rest of it will be the axon. These are going to be our sensory neurons in the PNS. Remember our peripheral nervous system. These will be the sensory neurons, or the neurons that are receiving information from from your skin, that kind of thing, and bringing that information back towards your central nervous system. Our last one is what we consider our kind of more traditional neuron, right? Our multipolar. You'll see it's got multiple dendrites, but it only has one axon, but it has more than one dendrite, hence the multipolar instead of a bipolar. These are going to be all of your motor neurons. So those efferent neurons that are taking information away from the central nervous system. So all of our motor neurons, as well as those interneurons, or those neurons that are located between other neurons inside the CNS. Okay, so that's kind of the basics of how our neurons are going to work and how they're going to conduct messages throughout our body. And like I said, we'll do some activities to help you reinforce this.